So I'm extremely happy to introduce the Insilico team, Petrina Kamya, and Frank Pun, of course, from Insilico Medicine, our main sponsors, but I guess also Alex. Without him, there was no conference. So please welcome to the stage. Give them a round of applause.
Maybe I can start first. Hello, everyone. Really happy to be here today to introduce our Pharma.AI platform and tell you how AI can facilitate the drug discovery, especially we can also focus on the aging research. Yeah. Okay. Sorry. Okay, so here is our Pharma.AI platform. So it is a fully integrated platform with biologies. We try to connect biology, chemistry, and in clinical. So basically, for the pandemics, we can use it for the omics data analysis, as well as the most important part is the target discovery. Okay, so I will cover the first part. So recently, we published a paper on trends in pharmacological science. Basically, we try to review the history of the target discovery. Actually, target discovery is very important because, let's say, imagine if we pick the wrong target, and then it will definitely affect the success of the drug development. So as you can see here, back to early 90s, for example, we have the discovery of mTOR as the target of rapamycin. And then recently, we applied the AI to help us to better to do the target discovery. And in last year, it's not only important to us, but for the whole industry. As you know, there are many AI-derived targets being validated with the wet lab experiment. This is a very important step to the industry. At the same time, the same year, we have the entry of the fully AI-decided drugs, AI-decided novel targets, and AI-decided novel molecules entered the human clinical trials. Now we are in phase 2. So actually, AI can help a lot of the early stage of the drug discovery, including the biomarker and target identifications, and as well as the indication prioritization as well. Of course, we can also apply the AI to help us to better design the novel molecules. Yeah. So here is the overview of our philosophies, how we can use AI to help us to identify the targets. For example, we have the omics AI scores. So basically, many of these models can calculate the scores in real time. So what we are doing is not just building a database telling you which target is connected to a particular indications, but it really depends on the data set we selected and we upload on the system to do the calculations. At the same time, we have our text-based KOL as well as the financial models to help us to run the targets. So overall, we have a very comprehensive overview of the target disease association. So basically, what we can do is we can start with the disease of interest and then identify the targets we want. On the other hand, we also can start from the target of interest. We find the best indications for that particular targets. That's very important because the indication prioritization, because many of us already have a very good target in our mind, so it is very important to expand the indications. And lastly, we can also do the biomarker discovery as well. So here I would like to give a quick update of our ARS project. So basically, just 1.5 years ago, we presented our data in the scientific conference like today. It should be the 7th annual meeting of ANSA ARS. Basically, we use Pandomics to integrate the publicly available data together with the ANSA ARS proprietary data, and then we find the targets. And further validated with the Mayo Clinics with this free fly model, And now we are already applied some of the one of the repurposing drugs on the ARS patients. Again, thank you very much for the support of Professor Bailu from 4B Technology, as well as Professor Fang from the Beijing University of Third Hospital. Yeah. And now I will show you a very quick screencast how we can pay up the targets.
ALS is a severe neurodegenerative disease with ill-defined pathogenesis and limited therapeutic options. Let's see how we can use Panda-Omics to search for therapeutic targets for this deadly disease. To start a project, we search for ALS on the main page. Here is the disease summary page where we can find most of the essential information regarding the disease. By hitting the Datasets tab, we can have a quick screen on the ALS-associated datasets available. All the data is processed in a uniform way. Clicking on the dataset name, for example, GSE 122649, will bring us to the dataset page where we can create comparisons. To create a comparison, we first classify the selected sample groups into either case or control, and hit Compare Selected Groups to initiate the calculation, which usually takes less than 5 minutes. Here is the summary page for the comparison report, including a volcano plot for differentially expressed genes, as well as a summary plot for affected pathways. Activated and inactivated pathways are categorized into corresponding cellular processes on the affected pathway page. The degree of pathway dysregulation was determined by the IPANDA algorithm. Pathways belonging to the same cellular process can be filtered by clicking the corresponding bar. For example, we are interested in RNA editing. We can find the expression profile and interaction network of all involved genes. By clicking each node, detailed information can be disclosed. And the same applies to the edges. To add the comparison into a meta-analysis for target ID, hit Add to Meta-analysis, and save in either an existing or a new meta-analysis. Before starting the calculation, we choose ALS as the therapeutic area. The calculation might take up to an hour to complete. Here is the Target ID page, where we can enjoy flexible therapeutic target hunting workflows. The main display is the heat map, illustrating the target performance in terms of the AI and bioinformatics scores. Panda-Omics predicts the target genes associated with a given disease through 23 omics, text-based, financial, and KOL scores. The platform also provides multiple filters and score customization to refine the target list to meet our research goals. To identify our proposed targets for ALS, we first select the comparisons for familial ALS as an example. Here we adjust the filters in a median novel manner. Switching on the druggable class filter yields actionable families of targets. All omics scores are selected, with the text-related scores disabled. ARDD2B, one of the unreported genes whose suppression strongly rescues eye neurodegeneration, is ranked as the top 20 target. Particularly, ARDD2B has outstanding performance in terms of the expression and heterogeneous graph walk models.
Okay, so now you can see how we can use our AI platform can find the novel target very quickly. So I will show one more case example. That is our recent paper in press from PNAS. Basically, we try to integrate Panda-Omics together with FieldDrop. So basically, we use our Panda-Omics platform to find the therapeutic targets using AI, and then at the same time, we use FieldDrops and then to assess the protein's probability for undergo these protein phase separations. Because you know the PPS is a very hot area, it will help us to discover some novel therapeutic strategy as well. So the idea is that we assess the potential targets targetability. At the same time, we assess the targets whether how likely it will undergo protein phase separations. Okay. For example, for each of the genes here, you can see that we will provide evidence whether it will have a sort of validated example for PPS, and how likely the protein gene is being located with the membrane-less organelles, and with the software, its predictions to undergo LPS, and also the pandomics ranking for this gene with the particular disease, and also the specificity of whether the gene is connected to that specific disease. And finally, we will build, we already built this potential target profile. For example, the disease in these promising priorities, okay, they are very likely to be benefits from the PPS-based therapy. So this is the whole idea. Actually, in this paper, we covered over 64 diseases and across over 10 disease groups. So it is pretty cool. And moreover, we also validate some of the potential candidates, whether they will undergo the PPS. For example, you can see that in two Alzheimer's disease cell model, you can see that for these genes and mouse genes, okay, with the addition of the A-beta-42 solutions, you can see this condensates. But after we added this 5% of HXD, which is chemicals that it will help to induce the lipid-lipid phase separations, you can see that the contents of these two genes, proteins, will decrease. So it implies that these two genes is likely to undergo the protein phase separations, and we can develop some interventions involved in this their concentrations to better to help with the Alzheimer's disease. So this is a very cool use case. And now I will pass to Petrina, who will tell you let's say once we identify the targets and then how we can move to the next step.
Thanks, Frank. All right. So Frank has shown you how we leverage multimodal data and AI to identify targets associated with aging. Now I'm hopefully going to demonstrate to you how we leverage other sorts of data and other sorts of technology in AI to discover small molecules that are optimized for different properties and also predict clinical trial outcomes. And I'm going to do this with a little story that's centered around the project that Frank did on ALS. So towards the end of last year, Alex and I were invited to Discovery on Target to present this work. Alex would talk about the target discovery portion, and being a computational chemist, I'd talk about the repurposed drugs, which is all fine and dandy. But then about 3 days before the conference, I had this fantastic idea. I'm also head of AI platforms. So I said, why don't we leverage all of our platforms and try to show how we can use them to come up with new molecules and predict the clinical trial outcomes based on the work that Frank did. More specifically, I wanted to work with the biologists to narrow down those 8 targets that were identified to 3, and then use Insilico, which is our platform for predicting the probability that a program will go from phase 2 to phase 3 successfully or fail, to predict of those 3 targets, which ones were the most likely to succeed in that phase 2 to phase 3 clinical trial. And then based on their predictions, then leverage our generative AI platform, Chemistry 42, to design molecules that had optimal properties. So I pitched this to Alex, and his response was like, wow, that would be cool. And I was like, oh, great. So now we have to do it. But the problem was we only had 3 days to do this. So I had multiple people to convince about my harebrained idea. So I first went to Frank's team, and I said, OK, guys, you have 8 targets. From a biological perspective, which of these 8 targets would be the easiest to prosecute? And Frank came up to me and said, KCNB2. That would be the best one. And I said, nah, it's an ion channel. A computational chemist, that's not something easy to go after. Give me some more. So he and his team came up with these 2 other targets, the ARDD2B and NR3C1. I was like, OK, these are more tractable. let me go to the Insilico team and tell them, based on these targets and the indication ALS, which of these two have a higher probability of being successful in terms of transitioning from phase 2 to phase 3. Insilico, for those of you who don't know, leverages multimodal data. We have 3 types of scores that are based on the trial design, target choice, and patient eligibility criteria that help you understand which of these programs, or your programs, are likely to make that transition successfully. In the end, you get a comprehensive report that not only tells you this probability of success, but also gives you a level of granularity regarding which features, for example, of your clinical trial are positively or negatively impacting that probability. Okay, so it went to that team. But the problem is that we only have a target and an indication. We don't have any clinical trial. But what we've found through our prospective studies is that just the target choice alone and an indication can give you a good enough idea of whether or not a program is going to succeed. And the target choice threshold of about, the score threshold of about 0.3, if you get that, that program is likely to fail. And if you get a target score, choice score above 0.46, then the program is likely to succeed in that transition. So of the 2 targets that Frank's team provided us with, the NR3C1 had a higher target choice score, 0.56, compared to the ARDD2023 target. So we went with the NR3C1 target. And we leveraged our Chemistry 42 platform to come in, to design molecules that are optimized for different properties. So for those of you who are not familiar with Chemistry 42, it leverages multi-parametric optimization to design molecules that are novel and diverse and have optimal properties. The types of parameters that you can optimize these molecules for include potency, safety, synthetic accessibility, selectivity, metabolic stability, novelty, and we also have the ability to predict the ADME-tox profiles of these molecules. What makes this platform unique about the data sets that we've used to train these generative algorithms? We have over 42 generative algorithms built into the platform, a very flexible reward function. We've recently integrated reinforcement learning with expert human feedback, so we get feedback from the community that helps us improve our models. And it's a very validated platform. We use this platform internally for our own internal programs. We have over 30. We've patented a lot of the technology, and we've been developing it for a while. But let's get back to the subject. So we were going to go after this NR3C1 receptor. I'll just call it GR for now. It's the glucocorticoid receptor. Some key things to know about this receptor. It's expressed in almost every cell in the body, which will mean selectivity is very important. And it's linked to therapy resistance across a variety of cancers. And there are known antagonists, of which there are 2. There's mifepristone. This is currently undergoing clinical trials in several oncology indications, but it has a lot of problems. It's very dirty. It inhibits a lot of CYPs, and it's not selective. However, there is another compound, ARDD2023, which is also a steroidal GR inhibitor, antagonist, but it has a slightly better profile than mifepristone. So there we had it. We had our target, we have our indication, we have a starting molecule, not too great in terms of its properties, but it's okay, it's good enough. So now we had to challenge the platform. Can we generate molecules that are novel, selective, restricted to the brain, because we're working with ALS, and have a better ADME-tox profile, predicted ADME-tox profile compared to ARDD2023. So this is what ARDD2023 looks like, just to highlight a couple of key things that we had to take into consideration when we were setting up the platform. The first is that steroidal core. That is what makes this molecule pretty promiscuous. It binds to the androgen receptor and all the other glucocorticoid receptors. So that's something that we had to tackle, come up with a core that's similar but not steroidal. Another aspect that we needed to get rid of is the aniline. This is genotoxic. Yeah, I think that's more or less it. But what was essential for binding is this isopropyl group in this position. So we encoded that as a pharmacophore feature. This carbonyl group, which is important for potency, and this alcohol group over here is also important for potency. So we use the Chemistry 42 platform, and I'll just quickly show you what that looks like to set it up. Pretty much in the same way, but I'm using a different example. You set up this structure-based design experiment. You upload the structure of your target. You encode that pharmacophore hypothesis that I spoke to you about, and basically just configure it. So tell the platform, I'm racing along because I know we're running out of time. Tell the platform that you want to generate molecules that have a better profile, so predicted ADME-tox profile, have a higher chance of crossing the blood-brain barrier, et cetera. And you let the platform run, and also novelty is important. That's a feature that you can select here. And after the calculation's been run, you get a database of 3D-generated molecules that you can rank and filter based on their properties. How well do they satisfy that profile? Okay, I'm just going to skip on. So that's what we did, and here's an example of some of the molecules that we generated. So this is a pretty potent glucocorticoid receptor inhibitor. It's a single-digit nanomolar. If we run it through our platform, that translates to a PLI score, protein-ligand interaction score, about 11.29. So the molecules that we generate, if we want them to have a similar potency, or predicted potency, or get that idea, we want to have a similar PLI score. These three compounds here are good examples of molecules that we predict would have a similar PLI score or activity to the auric compound. You can see their PLI scores are in the same order of magnitude. However, we've moved away from this steroidal core to novel cores. These are all novel molecules that are predicted to be more selective. And we actually did the screening against other glucocorticoid receptors, and we found that they score poorly in other glucocorticoid receptors. In terms of their CYP inhibition profile, these are way more improved than the auric compound. And if you were to look at the binding mode of one of the highest scoring molecules in the pocket, you can see that it maps very well with the auric compound. So this was, and this is it, I'm finishing wherever Morten is, I'm done. In this particular example, this was a fun story. We did this in 3 days. This is not how our asset team works. It also takes them a lot longer to come up with candidates, and everything is experimentally validated. But this is just to show you that it is possible to tackle target identification, this design of small molecules, and predicting clinical trial outcomes, leveraging AI. And we have over 30 programs internally, 3 of which are in the clinic, 1 in phase 2 and 2 in phase 1. And the rest are listed here. Happy to answer any questions, and thank you so much for your time.
Really an amazing platform. We have time for maybe one question. Maybe I can ask that question. And I'll challenging you a little bit, but have you considered biases in the data that you are using to train your models, and how do you deal with that?
Absolutely. So we make sure that our models, at least in chemistry, I can speak to that, are very well validated. The data sets that are used to train the models are very diverse, and we do a lot of cross-validation and test validation to make sure there are no biases in them. Yeah.
I think people are ready to go for dinner soon. So oh, do we have one more? Okay, quick question, very quick.
So when you generate your molecules in the chemistry part, do you have a synthesis route already provided for it as well?
We don't have a route, but we have a score. And this retrosynthesis-related synthetic accessibility score, what it does is it will break up the molecules that are generated. First of all, it gives the molecules a score based on how easy it deems these molecules to be synthesized. And what it does is it fragments the molecules into their building blocks. And if it can identify those building blocks in existing drugs, that lowers the score. And of all those building blocks that are used to generate your molecules, you'll get the CAS numbers. So you'll be able to know where you can purchase those building blocks and synthesize the molecules. No worries.
All right, thank you so much. Amazing platform, great talks.